everyone, welcome to our webinar today, which we're jointly hosting with Affymetrix. The title is Effective Utilization of FFPE Samples for Cancer Discovery. This is one of our customer spotlight presentations, and the speaker today is Dr. Joshua Schiffman of the Huntsman Cancer Institute at the University of Utah. Before we begin, I just want to remind everyone that this is a one-way audio, so you can hear us, but you're all on a muted line. But if you do have any questions during the presentation, please feel free to type them in as they come to you in the question panel on your GoToWebinar console there. Once Dr. Schiffman is done with his presentation, we will go to the questions and answer as many as we can. After that, there is a 3-question poll we'd like you to participate in, so if you could just take the time to do that, it will take about 2 minutes. That would really help us out. And once the webinar window closes, another browser window opens up with an area where you can type in any additional questions you may have or any feedback or comments you may have for us, so please feel free to do that as well. Before we get started, I just want to hand it over to Padma Sundar of Affymetrix. She's the product manager of OncoScan, and she'd just like to say a few things about the OncoScan service. Padma?
Hello, actually, this is Gianfranco De Feo from Affymetrix, and I'm standing in Padma's place. First, I would like to welcome everybody to this conference call, and thank you for joining in. And I would like to thank Dr. Joshua Schiffman for presenting this presentation to you today. Affymetrix offers the OncoScan product for FFPE samples to use for a variety of different research functions, especially with FFPE samples for various cancer discoveries. You will hear some very interesting results from Dr. Schiffman today, and would love to hear any questions that come in regards to the products or the utilization of the products. We also recently are offering the OncoScan services together with the biodiscovery tool that allows simple analysis of the results that come from the OncoScan products. So with that, I will turn it back over to Shalini. Thank you.
Thank you, Gianfranco. Now I just want to introduce people to biodiscovery if you're not familiar with us. We provide software and services for genomic data analysis from high throughput technologies in the areas of copy number, expression, and image analysis. Over our history, we've developed several products which stem from our basic philosophy, which is just outlined here. We try to use the best software engineering practices to develop robust, efficient, and elegant platforms. We use the well-accepted analytical approaches that are available out there rather than inventing our own algorithms. We focus on the software engineering. And we also try to develop single button solutions for research scientists where they can easily go from their raw data to results without the need for programming skills. So one of our products is biodiscovery Nexus Copy Number, which is a platform-independent solution for copy number analysis from CGH, SNP arrays, as well as NGS data. It's simple and powerful and allows users to go from raw data to discovery very quickly. And also recently we've added numerous cancer-specific algorithms. And also in light of our tight partnership with Affymetrix, we've added support for OncoScan data, where the data generated by the OncoScan service can be loaded directly into Nexus Copy Number. We've also optimized parameter settings and have added several tools for correcting the data, such as wave correction for the GC content. We also have support for the ASCAT algorithm to automatically determine sample ploidy, as well as the % normal cell contamination levels. Also, the software allows you to integrate data from any other array type as well, including Affy 500K, SNP6, Cytoscan HD with the OncoScan data, so you can look at everything all in one frame. And then we also have complete downstream analysis and visualization tools, and you will see some of that in today's presentation. And before we begin, I'd just like to introduce Dr. Schiffman to everybody. He is an associate professor in the Division of Pediatric Hematology Oncology at the University of Utah, and he's also the medical director of the Pediatric High-Risk Cancer Clinic at Huntsman Cancer Institute and is an investigator at the institute. Dr. Schiffman began his medical training at Brown University and then moved west to complete his training at Stanford University. He's trained in pediatrics and pediatric hematology oncology. 4 years ago, he joined the Huntsman Cancer Institute at the University of Utah, where he studies pediatric hereditary cancer syndromes, with a special interest in genetic susceptibility to childhood cancers. Much of his research focuses on copy number changes and studying both genetic risks for cancer in patients, as well as the genetics of specific tumors once they develop. He has been successful in the discovery of genomic signatures in childhood acute lymphoblastic leukemia and Ewing sarcoma that are highly predictive of clinical outcome. He has made great strides in effective use of the challenging FFPE samples in his discoveries, and he will be presenting some of this work here today. So now I'd like to hand it over to Dr. Schiffman so he can begin his presentation. Dr. Schiffman?
Out there. All right. Thank you for those introductions, and I think we'll go ahead and get started. Hopefully you can see my screen out there, and you are comfortable. So sit back, relax, put on your thinking caps, and for the next 45 minutes we are going to explore FFPE and some of the new technologies that can help us to interrogate these paraffin samples for cancer discovery. So for this morning or this afternoon's talk, depending on where you're located, we're going to discuss this technology called molecular inversion probe, and it's offered through Affymetrix. It's called the OncoScan FFPE Express Panel. We'll get into more details throughout the talk about how this technology works. We'll focus specifically on acute lymphoblastic leukemia, Burkitt lymphoma, and Ewing sarcoma. And the third objective, which will be sprinkled throughout the talk, is really to understand how these molecular and genomic findings we're making in the laboratory can be applied to our patients to improve prognostic risk stratification and outcome. So with that, let's get started. First of all, the cancer challenge. So as many of you know out there who are listening, cancer is the leading cause of death worldwide. There are over 8 million deaths per year from cancer alone. And in fact, 15 people will die from cancer every minute. That means by the time this webinar is over, there will have been nearly 1,000 people who will have succumbed to this disease. Half of all men and a third of all women will get cancer throughout their lifetime. So cancer is out there, and it's a problem. Now for me, as a pediatric oncologist, it's a particularly challenging problem because cancer is the leading cause of disease-related death in children. One child who dies of cancer is one child too many. And what we hope is through the study of genomics, we can increase those odds and improve survival of young children and adults with cancer. At the Huntsman Cancer Institute in Salt Lake City, Utah, we focus on cancer genetics and how can we apply all of the surrounding studies, population genetics, pathways, preclinical, and even clinical studies to apply to the individual patient, to come up with a personalized oncology plan, or individualized oncology plan, as some people are calling it. And really, what that means is that we take all of this information and we use it for each individual patient to focus on how we can get the best results. And for any of you who were at the recent American Society of Clinical Oncology, or ASCO, meeting just a few weeks ago, you would have noticed that many of the talks were focused on how we take this genomic information and use it for our individual patients. Now, one thing that we have in our advantage are paraffin samples. So I don't know how many of you out there listening are pathologists, but what I can say is I am not a pathologist, but my closest friends and literally neighbors are pathologists. And pathologists are wonderful for a number of reasons. One of the main reasons being they are incapable of throwing anything out. And that really works to our advantage. Every sample that is removed from a patient is saved, is embedded in wax and paraffin, and is then available for analysis. So that means there are now over 1 billion tissues, as quoted in this Nature article, and we know if it's in Nature, it must be true, right? That 1 billion samples are now available for analysis. And these are an incredible opportunity for us as researchers and clinicians, but they also represent a huge challenge. During the paraffin embedding process, the formalin actually cross-links the DNA. That makes it very difficult then to interrogate that DNA once it is extracted and deparaffinized from these samples. However, this is such a treasure trove of data because it is so clinically annotated, meaning we know how the patients presented. We know how the patients did. We know all of the clinical information associated with these paraffin samples. So if we could just figure out how to analyze those paraffin samples, we would be well off. And in fact, there is a way to do it using SNP microarrays, and that technology is called molecular inversion probe technology, or MIP technology. Again, that is now offered commercially from Affymetrix as a product called OncoScan FFPE Express. So let me take a few minutes to explain how this MIP technology works before we actually delve into some of the clinical examples and laboratory examples. This is about 180 base pair oligonucleotide. These red regions right here are designed specifically to match up and bind to your region of interest on your startup DNA. You only require a small amount of DNA, 75 nanograms, although we have gotten this to work on as low as 3 nanograms of DNA. These probes bind to your DNA, to the area of interest, and there is a gap here, as illustrated by the animation, which is eventually circularized by the missing SNP when the probe is bound. There are different areas of cleavage sites built into the probe. There are PCR primer sites so that once bound, after an exonuclease step that chews up all of the unbound probes, the bound probes are then cleaved and amplified. And most importantly, this unique barcode sequence built into the probe is what binds to the microarray chip. This is really very important and is the secret to this assay's success. Why? Because normally with SNP arrays, it is the template DNA, the startup DNA, that is amplified and bound to the microarray. Well, you've all heard the expression garbage in, garbage out. If you amplify DNA that's been cross-linked and that is in poor quality condition, that DNA is going to give you poor quality results. Also, the DNA is going to have many regions that cross-hybridize to other regions in the microarray, and that's going to provide more noise in your signal. With this technology, though, the reason I like it so much is that it's not the DNA here that's being amplified. It's actually the probe itself. And so at the end of the day, it's this non-human barcode sequence, which avoids the issues of cross-hybridization because it's a unique sequence. And it's this non-human sequence that binds to the microarray. So even if you're working with really poor quality data, or poor quality DNA rather, old samples that have been cross-linked, that have been stored in paraffin, it no longer is such an issue because it's not the DNA that's being amplified and bound, it's actually the probe. And in the latest version of the OncoScan assay, there are 334,000 probes. They also not only include probes for copy number analysis, but also somatic mutation information. And we can talk about that more during the question and answer period if people would like. And the probe assay has been designed so that it focuses on cancer genes. So you have fantastic coverage for all of your oncogenes and tumor suppressor genes, any cancer-related genes. But it also has been designed so you have good coverage throughout the backbone of the assay, so you get genome-wide data like you're going to see in a moment. The advantages of this particular MIP technology is you can choose any non-repetitive sequence of interest. So these regions that bind to the DNA are custom designed like we were talking about just for the cancer genes. You have reduced DNA requirement, so 75 nanograms compared to usual 500 nanograms or more, We talked about the DNA quality being less important. You have quite a dynamic range. So you can detect amplifications cleanly and clearly up to 60 or above. We have some examples of 80 or higher. And it has very high sensitivity and specificity. So your data accuracy is greater than 99.6%. Well, this is where I'm sitting now in my office. It's a beautiful day here in Salt Lake City looking out at the mountains. And what we do here is we have one children's hospital. This children's hospital covers this entire Intermountain region. And what that translates to is only 170 new pediatric cancer diagnoses per year. So these areas aren't well populated, but they are quite beautiful. However, if we want to study cancer, that means that we only get 30 to 40 new leukemic diagnoses per year in children, 6 to 10 Ewing sarcoma, 3 to 5 Burkitt's lymphoma. So if we did a prospective study, it would take us quite a while to get enough data to say anything meaningful. However, now that our friendly pathologists have saved everything in paraffin and have been doing that for the past 100 years, we can go into our archives, we can go to the paraffin warehouses where these FFPE blocks are stacked and stacked and stacked, pull them out, and do our molecular analyses. So let's get started. So ALL, acute lymphoblastic leukemia. This is the most common childhood leukemia that we see. It is present in over a third of all of our pediatric cancer diagnoses. Traditionally, we do quite well with this. We have a cure rate of 80 to maybe even 90% of patients. However, those 10 to 20% of patients that relapse, unfortunately, often will go on to succumb to their disease. We need to do a better job understanding where does ALL come from and what are some new targets that we can identify to help treat this disease. Here we see a visualization of our first experiment. We took 60 patients using paraffin-embedded aspirate clots that were 5 years or older. We extracted the DNA. We shave off the wax of these samples. We take out the DNA, and then we run it through the Affymetrix MIP, or OncoScan assay. Currently, the OncoScan is offered as a service product, meaning we send the genomic DNA to Affymetrix after we prepare it in the lab, and then we have the data returned to us. Although it is being prepared for an off-the-shelf product, meaning you can run it in your own research or clinical laboratories starting, I believe, in 2013. For these experiments, though, we did send them to Affymetrix, and this is the data that we got back. This is visualized on the Nexus browser, which we found actually is one of the best software programs available for analyzing this MIP data. BioDiscovery has worked closely with Affymetrix to make sure that the data is returned in a form that can be readily explored and analyzed. And so here we see an example of that. So these are the summary of all the chromosomes. Our 60 individual samples, the data has been pooled by the Nexus software browser. And what you can see is the percentage of samples indicated by the length or width of these bars indicate what percentage of the samples actually have that amplification or deletion. Here, green is a gain and red is a loss. And so what you'll notice right away is there are quite a few trisomies that have been described commonly in leukemia, 4, 6, 10, 14, 17, 18, for example. And here's 21. You'll notice that the Nexus browser colors this in darker green, which indicates that it's not just a trisomy. Here we have examples of patients that have high gains, so copy numbers 4, 5, 6, or even higher, and again indicated by the green bars. You see some spikes in the red bars. These are the known areas of deletion. Here, for instance, is CDKN2A or P16. We know that that 9P20 region is commonly deleted in ALL as well as in other cancer. And again, you see the very focal deletion there. Well, when the data was returned to us, we took a careful look at it. Here again is the same summary of all the data. Here are the individual patients. And what we found was that over here on 22, there was a spike, a focal deletion. And this was in a gene called VPRB1. And here again, you see the individual patients have this deletion in red of this region. And so we found that in a quarter of patients. And when we looked closely at that VPRB1 gene, what we learned is that VPRB1 combines with lambda-5 to form what is called the surrogate light chain. So this will take you back to your days of immunology. And if you'll recall, the surrogate light chain, its job is to hold the place on the immunoglobulin heavy chain receptor as the lambda light chain undergoes rearrangement. And the bottom line is that this VPRB1, you need it in order for the B cell to continue during normal development. Without VPRB1, the B cell cannot go on to become a mature and functioning B cell member of society. Or, as I like to say it, without VPRB1, the B cell can't have its B cell bar mitzvah, right? You need VPRB1 for the B cell to go on and mature. Why is that important? Well, we know that leukemia is really B cell that is stuck in this immature or pre-bar mitzvah state. The B cell is young, and that really is what cancer is. So this was a very intriguing deletion to us when we found it. We knew, though, from looking in the literature that VPRB1 actually had been reported before. And previously it was considered a normal consequence of lambda light chain rearrangement. We'll talk about that in a moment. But interestingly, this VPRB1 had been reported to be associated with high-risk samples. Its underexpression, which would go along with its copy number deletion, had been associated with relapse and worse outcomes. So you may be wondering, well, Josh, what's the big deal? You found something that's already been described before. Well, that's true to a certain extent, but we wanted to look carefully at this concept of the VPRB1 deletion being part of lambda light chain rearrangement. And the reason that people had thought that was that if you look at VPRB1's location, it's actually located right smack dab in the middle of the lambda light chain among all of the other variable cassette regions. Again, if you think back to your immunology days or your immunology classes, you'll recall that the reason we have such a large immune repertoire is that the V chains and the J chains rearrange to form a unique epitope or a unique region that can identify novel threats to our immune system. And the way that works, if you look on the bottom here of this figure, you see all the V representing the unique variable cassettes. Here's the J region, here's the VJ junction, and here's our friend VPRB1 sitting around minding its own business, located by perhaps a fluke of evolution right in the middle of these variable regions. Well, the way that VPRB1 was thought to maybe get deleted along with these variable cassettes was by the RAG gene, through the magic of PowerPoint and animation. Again, I'll show you again. Here's RAG1. It comes in, and it basically splices out everything between and brings this variable region here. Hopefully you can see my little pointer and your home locations there, or your office locations. It brings the V cassette to the J over here, right? So it splices everything out one final time, comes in, knocks out everything in between, including VPRB1, so V goes close to J, and that's why people said, well, you know, VPRB1, yes, it's deleted in leukemia, but it's probably because during rearrangement it gets spliced out. We wanted to look at that more closely, and actually this is where Nexus really was a great help to us with its visualization. If you look here, here is something called mature B cell leukemia or lymphoma, Burkitt's lymphoma. These B cells have already undergone B cell differentiation, right? They already had their B cell bar mitzvahs. They're mature, functioning members of B cell society, and then they turn into their cancer afterwards. So VPRB1 has already been deleted if it's going to be deleted. The light chain rearrangement has already occurred. These are mature B cells. And if you look here, you'll see here's the VJ junction, and there's a contiguous deletion, right? RAG gene has come in and deleted everything in between, has recombined it, spliced it out, and brought this J junction close to this V junction. Again, you can see in this Nexus browser here that here's the individual, here's the red deletion. Well, we did find one sample in the mature Burkitt cell lymphoma that had a VPRB1 deletion. This VPRB1 was located, just as people predicted, in the middle of the variable regions, and it was deleted clearly as part of this rearrangement. So here's the exciting part. When we looked at leukemia, what you could see, it was a very focal deletion. This VPRB1 deletion was not part of this VJ recombination of the RAG mechanism. right? You can see the contrast between them. Here's the Burkitt cell, very clean splice site right here in the VJ junction and a contiguous deletion, whereas here it's very focal. So that was the VPB1 deletion right there. And this was interesting because if we looked at our cohort of patients and we looked at other B-cell developmental genes, we found these were also deleted in B-cell leukemia. But actually, of those, many of them only had VPB1 by itself. So at the end of the day, nearly a quarter of our samples in pre-B ALL had just the focal VPB1 or its enhancer or slight deletion in front of it, was the only B-cell developmental gene deletion. So what we think we may have found is a new subset of leukemia or a new mechanism for how leukemia may form, and this now can offer potential therapeutic targets. And we've gone on to confirm this deletion using other data that's available from other microarrays besides the MIP platform, samples from the Children's Oncology Group, from St. Jude's Children's Research Hospital, and find about the same number of samples have this focal isolated VPB1 deletion. So that was one of the interesting things that came out of our study using paraffin blocks, archived samples, and Nexus Browser to make these discoveries. We'll next turn our attention to Burkitt's lymphoma. This is a very aggressive cancer. It's one of the fastest growing cancers known to mankind, a doubling time of 24 hours. This is a cancer you want to catch right away as it's growing. But because it grows so fast, it actually is fairly responsive to chemotherapy. However, there are about 10% of patients with Burkitt's lymphoma that are refractory and or recur, and those patients are very difficult to treat. They often go on to die. And we need to figure out what's different about those patients that relapse versus those patients that don't. So we set about to doing that, again, taking the same approach, taking paraffin blocks. This was done with our hematopathologist colleague, Dr. Rodney Miles, who's here at the University of Utah. And we were able to collect 32 paraffin-embedded biopsies that had at least 80% tumor. We were able to extract both their genomic DNA and their RNA. And we were able to get clinical data on 27 of these patients. Importantly, with solid tumors, which lymphoma is considered a solid tumor, we were able to get the surveillance bone marrows. With solid tumors, we actually look quite closely at the bone marrow to make sure that disease hasn't spread or metastasized. If it hasn't, and that bone marrow is normal, it is still saved in those paraffin-embedded aspirate clots that you see at the bottom. So that gives us a unique opportunity to look to see if the genomic changes that we're finding in the assay are actually somatically acquired, so unique to the tumor, or if they're germline and they've been passed from the germline to the tumor. So for this study, we had 27 marrow clots and paraffin samples on 32 patients that we had clinical data on for 27. So we went ahead and extracted the DNA, ran the OncoScan FFP Express platform with the MIP technology, and here's what we found. A quarter of our patients had a gain of 1Q. Now here you see those 7 patients zoomed in up close. This is, again, a nice visualization by Nexus, which makes it very easy to manipulate the samples to really explore it further. And you can see, based on, here of those 7 with this gain, 50% had a gain in this region, and you look up here and you see this is nearly 100% of the patients had a gain right here. So again, interesting genes to go out and explore. Here we saw a loss of 17p, and again, using the visualization tools, you can really hone in quite easily and quite quickly to explore what are the common overlapping recurring regions. And finally, here we found nearly 10% of the patients, just over 10%, had a very focal gain in this 13q region. Like we were discussing before, this dark region right here, that's very exciting. That means that there's a high gain right there. So we said, let's explore that a little further. Here is, again, another way of visualizing the data using Nexus. And what you can see is each of these dots is one of the probes across the OncoScan platform. So clearly, even though it's cancer-centric, you get great coverage of the entire chromosome. And when you look here at 13q, what you can see is there's a high gain focused right there. Again, a high gain indicated by the double green bars. One green bar means there's a trisomy, but two green bars means there's an increased copy number. And you look at this second sample, again, a very focal gain right there. And again, the third sample, another high gain right there as well. So when we looked closely, what we saw was that these were the genes involved in those high gains, and that became very interesting to us because there was a microRNA here, microRNA 1792. And if you go to the microRNA libraries and you look that up and you do your PubMed search and you do your due diligence, what you find is microRNA 17 is quite interesting because it's associated with many different cancers and may be considered an oncogene. We had the RNA available from these paraffin samples, which were 10 years old, and we were able to extract that RNA, and we were able to look and do a TaqMan assay and actually show that those patients that had the gain had an increased expression of that microRNA. So it's not just a copy number gain that's sitting there looking pretty, it's actually doing something. And what it's doing is it's increasing the expression of this microRNA. Here are the samples that didn't have a gain, and you can see their expression was much lower. Well, because we're clinicians and we're interested to see how this actually affects patient care, what we were able to show was that of those patients, even though there was a small number of them, only 3, 2 of 3 of those patients with that gain were the 2 of 3 that relapsed. And whereas one of them had that gain, it was only 1 of 21 that didn't relapse. And that still came out to be statistically significant, or at least suggestive. And so now, because we can work in paraffin, we're working with collaborators across the country. Any of you out there listening are welcome to join along, and we're able to collect samples, these rare relapse samples, and even though each institution may individually have only 2 or 3 samples, if we all work together, we could get 50 samples, maybe even 100 samples of these rare relapse Burkitt's lymphomas, the lymphomas that kill these children, but we can now study them. Why? Because we can use paraffin. We can use these archive samples, and we can figure out what are some of the other changes that are going on. So that is very exciting to us. And again, I'll direct you to the British Journal of Hematology, where we published these findings last year. The last cancer we're going to talk about this morning, and again, I can't see any of you out there, so hopefully you're eating your lunch and still awake and paying attention, but we'll go forward nevertheless. Ewing sarcoma, this is the last cancer we'll talk about. This is a very interesting cancer. This is quite rare. It only occurs in 200 to 400 people per year in North America. It makes it quite difficult to study. They all look like this small blue cell cancer under the microscope. It occurs in the bone and the soft tissue. Now, what's particularly interesting and challenging about this cancer is we know if you present to our pediatric oncology clinic with localized disease, meaning you come in to see us, you have one lump, say, in your rib, that's it, no other cancer anywhere in your body, you have about a 75% chance of survival. We learned about a decade or so ago that if you increase the therapy, if you add additional ifosfamide and etoposide and space the therapy closer together, if you have non-metastatic localized disease, you can increase the cure rates from about 55% up to 70% or maybe even higher. So that was actually a nice thing to see for our patients. However, if you come to our clinic and you have metastatic disease, meaning you have disease in more than one location, you have a tumor in your ribs, and maybe say a tumor in your pelvis, we know that unfortunately no matter what therapy we add, no matter what we try to do, we cannot budge this curve. Your actual survival rate, if you have metastatic disease in Ewing sarcoma, may be as low as 10%. What that means is these young adults are coming to see us, these adolescents who are in the prime of their youth. If they have metastatic disease, we know that within 5 years, the majority of these patients are going to be dead. This is like a death sentence. This is a disease we need to better understand. It looks, like I said, just like this under the microscope. So it's all the same. But we know that there must be a molecular difference going on, something that's driving metastasis. And our challenge as cancer researchers and scientists and clinicians is to figure out what is the molecular basis for this difference in disease. If we can do that, then perhaps we can more accurately risk stratify patients and come up with novel therapeutic targets. So taking the exact same approach, we worked with our pathologists, we dug through the warehouse of FFPE blocks, and we found 40 primary tumors, which may not seem like a lot to you out there at home or in your office, but Ewing sarcoma, this is actually quite a large study. This is actually a very respectable study of 40 tumors for a low or a rare cancer rather, but we were also able to find metastatic lesions. That was so important because often metastatic lesions, you can imagine if a patient's coming in to see you, right, if you know they have primary disease, you're not going to biopsy the metastatic lesion because you already know it's metastatic and it's not going to give you any additional information. However, there are rare situations where during the course of the year, maybe 1 or 2 patients, you're not sure if it's a metastasis or an infection. So the surgeons go in, they biopsy it, and sure enough, it is unfortunately a metastatic lesion, but it's saved in paraffin. So what that meant was going back 12 years looking at our data, we were able to actually find 12 metastatic lesions to help analyze. And what I tell people in our lab is that no one dies of primary disease. It's the metastatic disease that kills you. So this is really where we need to focus our efforts. Similar to the Burkitt lymphoma study, we were able to get our surveillance bone marrows that were normal, and we obtained 31 of those. And again, these were samples that dated back from 1994 to 2010. We were able to get the clinical data that was associated with these paraffin samples, most importantly, event-free survival and overall survival. Here is, again, Nexus visualization, a summary of all of the changes of the 40 primary tumors. We found things that had been described before, like these 1q gains and trisomy 8. Here's a deletion of 16, which we found as well. But one thing that particularly caught our eye was this gain here on chromosome 20. Now, as indicated, if you squint your eyes looking at your computer screen, you'll be able to hopefully see that this falls within this green bar here, which is Nexus's way of saying it is a trisomy. But if you look right here, there's a gain. It pops up. So right here is a gain of 11. So copy number 3, trisomy 3, and then boom, 11, and then back down again. Well, this is really very an interesting finding, and so we wanted to see what was going on with that. But another thing to also point out just quickly is that you'll see that you'll have a change in the allele ratio here. This data comes back with allele ratio information, and so again, just confirming and validating this finding. So what did we see? Well, over here indicated in the dark blue is a gene called CBPB, and our eyes were drawn to it, not because it was dark blue, but because of what it actually does. So look here, CBPB, this is a CCAAT enhancer binding protein. Most importantly, this is involved in the expression of collagen type 1 gene. So we said, hey, this is Ewing sarcoma, this is a bone tumor and a soft tissue tumor. Collagen is right there in the middle of it. This may be an important gene. What we wanted to know was, did this copy number change actually translate, though, into anything biologically significant? You can have copy number changes, and they may just be passengers. They may not be driving the tumor or have any difference in expression. What we were able to do was actually find that if we did immunohistochemistry staining for the protein of CBPB, we found that those with 2 copies, normal diploid, had variable to low diffuse expression, indicated in brown of CBPB. When we looked at other samples that had 3 copies, here you can see it's starting to get darker expression. It's a bit more nuclear in this sample. And over here, here's our friend with 11 copies. And what you find is, look at this dark nuclear staining. This looks as though my 3-year-old took a brown Sharpie, and instead of coloring on the walls like she usually does, she actually scribbled here on all of these cells. Look how dark that is. So what that means to us is that there is biological significance. This gain here of 11 copies actually translates into an increased expression of this protein. But then, because these were paraffin samples, because we had outcome data going back 12 years, we could take the next exciting step of actually seeing, does it make a difference clinically? And if you look here at event-free survival, what you'll see is those patients, the 6 patients, not too many, but those that had the gain, had a much worse event-free survival compared to those that were normal. And if you looked at overall survival, wow, look at this. All of those that had the gain actually drop off and die. Now, even though these are small numbers, n equals 6, it's still statistically significant. So this has led us now to go on and further explore CBPB as a potential therapeutic target for Ewing sarcoma. We also, along with the other genome-wide data, were able to find, using the Nexus algorithms, those regions that were statistically significantly associated with worse outcomes. So here are our 8 regions. Some are moderately associated with worse overall survival. Some are really significantly associated. What we wanted to ask was, if a patient comes in on day 1 and we analyze their sample with Oncoscan or any other type of array platform, can we use these regions to actually predict outcome? So we divided up the patients into 2 categories. One category had no copy number changes. We called that the multi-factor copy number, or MCN index negative, so 0 changes. The other was an MCN positive index, meaning they could have at least 1, maybe 2, maybe 3, maybe all 8 of these changes. So it's 0 versus 8. You can't see me waving my hands in my office, but I'm getting very excited about this. So you have 0 copies in one category. You have 8 copies, at least 1 up to 8 copies in the other. How do these patients do? Well, here's what we found. If you had 0 of 8 of those changes, your event-free survival was pretty good at around 80%, meaning only 20% of these patients relapsed. If you were positive, though, it was closer to 60% of patients that relapsed. But what we really care about is overall survival. How did these patients do? How did these young teenagers do in the long run? Well, look at this. 100% survival. So we had 100% overall survival of these patients who were multi-factor copy number index negative, right? Versus only 40% survival if they were positive. That is a huge difference. Moreover, this study was done over the course of, took us only 9 months to do the study, but we have data going for 12.5 years. So this is 12.5 years of survival data. This meant that if we wanted to do this study starting today, it would take us 12.5 years to get the same data. Because we can now use Paraffin, we can go back in time, and like I said, the study only took us 9 months. It took us 1 month to get the genomic data back and prep the samples. It took us 8 months to get the clinical data. So now we can do these studies with the billions of samples that are located around the world and get our results very quickly. Just winding down here, something else to quickly show you. We were able to take those metastatic samples and really explore this concept of metastatic progression. Here's our primary tumor. The traditional thinking is you have a daughter cell with a subclone. That subclone goes on to seed somewhere in the body, and then you have these metastatic, which are related but different from the primary tumor. We can actually look at these genomic signatures, and we can see here's the primary tumor, here's what's gained in the metastatic lesion. Here's another example of the same thing. Here's our primary tumor. Here are deletions. Here are our gains. And we look at the metastatic sample, and we see over here that there is a deletion in a certain population of cells within this metastatic lesion. Just like Burkitt's lymphoma, we're now working with colleagues and collaborators, and welcome all of you to participate, collecting these metastatic lesions, these rare metastatic lesions, and if we can analyze 50 of them or 100 of them together, looking at their genomic changes, we may find out some of the unique changes that allow metastasis to occur in patients, and also, importantly, come up with novel targeted therapy. So far, we've run Oncoscan on colorectal cancer, brain tumors, melanomas, paragangliomas, GIST, ovarian tumors, renal cell carcinomas, and rhabdomyosarcomas. And we are continuing to work with paraffin samples, and we're getting really high quality results. As an example of this, and again, this is the last part of the talk here, we had a collaborator here in Utah who heard us talking about these paraffin slides and said, do you think this would work on the glass slides that we all have saved in our offices? Many of those glass slides have paraffin on the cover slips. That's what they're made out of. Do you think it would work? We said, sure, let's try it. We'll throw the samples in right next to the Ewings and the Burkitt samples. So we were able to get slides that were 8 to 10 years old, sitting in this collaborator's file cabinet in her office, minding their own business in the dark until we got them, pulled them out, scraped off the cover slips, extracted the DNA, and ran them. And here's the data that we got back. Really exquisitely clean results for SNP array, regardless of the fact that it was paraffin and 10 years old, sitting in a file cabinet. And here you can see very clearly gains and losses of these lymphoblastic lymphoma samples. Here is another example. If we zoom in, what you can see, again, each of these dots are our molecular inversion probes. Here's a very clear clustering of the probes, a very nice example of a homozygous deletion in this paraffin slide that was 10 years old. Here's another example of a homozygous deletion right next to a hemizygous deletion next to diploid. So in this one slide, paraffin-embedded slide, what we find, 10 years old, we find diploid, homozygous, hemizygous very clearly delineated. Here's another example of a sample that technically failed, but even though it failed, you can see a little bit of noise. Using the allele ratio information, using the Nexus algorithms, you can very clearly pick up these deletions, again, in slides 10 years old. So finishing our webinar for this afternoon, what I can tell you is that copy number changes correlate with clinical outcome in childhood acute lymphoblastic leukemia, in Burkitt lymphoma, in Ewing sarcoma, and many other tumors, too, that we haven't had a chance to talk about together today. These discoveries may lead to novel therapeutic targets for development, as well as understanding of where these tumors come from, how they arise. However, success depends on clinical collaboration and access to patient samples. Getting this clinical data is key to say something meaningful about these samples. But the good news here is that paraffin is no longer a barrier to this type of research. But with that, I will end. I will thank all of the multiple collaborators and colleagues who've helped with different aspects of this study, the funding mechanisms, but most importantly, the patients and their families who've allowed us to do this type of research. And before I end, I'll just say my email address is up here. I've added this since this morning's webinar. So here's my email address, and please feel free to contact me if you have any questions that we can be of any assistance with in your own research endeavors into cancer. So with that, I'll stop, and I'll turn it back over to Shalini, and I hope we have some time for some questions.
Great. Thank you very much, Dr. Schiffman, for this wonderful presentation. We're going to set aside a few minutes for some questions, and then we'll open the polls. Here's a question for you, Dr. Schiffman. Well, we are getting deletion and duplication. What about other missense, mutation, and indels? Perhaps they would be the driving mutation, and all you are seeing are progress in events.
Right. So there's a couple of different answers to that question. So absolutely, it's important to recognize what's a driver and what's a passenger. And one of the ways you can do that with this type of data set and using some of the visualization tools that Nexus offers is you can look to see what percentage of your tumor actually contains a deletion or an amplification, what percentage of the allele ratio is actually involved in that change. So you may have one sample where only 50% of the sample actually has a deletion, which again you can pick up from the copy number data and the allele ratio information. But then there are other regions of the tumor, and they may be your smaller indels that can be picked up depending on what the spacing of the probes are in that particular area, which may be involved in 100% of the tumor. And if they're involved in 100% of the tumor, that would suggest that that was an early event that happened in the evolution of the tumor. And because it happened early on, it may be more likely to be a driver. In terms of the mutation data, I mentioned this earlier on, one of the nice things about this platform is that it also includes mutation data to some of the most common somatic mutations that occur in tumors. And you're able to get that mutation data back along with the copy number data. and you receive a score that tells you how likely that mutation is present, and then you can go on and do more focal validation of that mutation. But that becomes very helpful in actually identifying some of these missense mutations that, as the audience member pointed out, may be some of the drivers of the cancer.
Okay, thank you, Dr. Schiffman. Another question here is, how does this technique compare to next-gen sequencing? What are the advantages?
So this is always an interesting question and comes up quite a bit. And, I don't think that one technology is going to be the sole answer. I think both of these, this type of technology along with next-gen sequencing, are going to be used together to find out information. However, that said, right now, at this point in time, next-gen sequencing can give you some results in paraffin, but the quality is not quite that good yet. And I think now is sort of a window of time where this technology really fills a void and can do samples that are, 20 years old. I think some of us are working on getting samples 50 years old. So you can get large cohorts of clinical data that you can't get with next-gen sequencing. And I think that will be one of the advantages, this ability to work so well in paraffin.
Thank you. Another question here is, how much of this data analysis you've shown was done with the Nexus software and how much was done with other packages?
For the data that I showed today, really almost all of it entirely was done with the Nexus software. We do have other projects where we have other data sets that we can merge using biodiscovery platforms, but we have our own homegrown statistics and biostatisticians. But we use, though, and this is important, we use the output data that Nexus generates for us to take the genomic analysis and do other types of investigations with it. So the data I showed today, though, all of it was done using Nexus. One of the, and again, I don't work for Nexus, and I should defer a little bit to the Nexus folks to speak in more detail, but there are many different algorithms that they incorporate, so publicly available algorithms, so wave correction and ASCAT and other types of techniques that have been described in the literature, and that gets imported into the Nexus browser, so you can use your favorite analysis program with Nexus. One of the nice things about using Nexus that I have found, and again, I don't work for Nexus, so this is just as my own opinion as an independent investigator, is that they've been extremely responsive to any questions or issues that arise in the analysis. And more importantly, if we come up with a technique or if there's an analysis approach that isn't offered by Nexus, they are very responsive as a company, more so than we've found with other companies, to actually incorporate that into the Nexus software package so that you can go forward and use all of their programs together. And it's been very helpful for identifying clinically relevant data.
Okay, thank you. Have you used your FFPE samples on any other platforms, such as Agilent, ACGH, AFISNIP6, Illumina, Infinium, etc., and compared it in Nexus?
We have not done head-to-head comparisons, but we've talked to other investigators who have, and what we find is that the SNP arrays that are out there, specifically SNP 6.0 and some of the other arrays, they work fairly well actually on new paraffin samples. So paraffin blocks that are a few months old up to a year old, you get really pretty good data. However, once you get past 3 to 5 years, the quality of the data rapidly degrades because the DNA rapidly degrades. The other thing that's interesting is we find we have some examples of fresh samples with SNP 6.0 compared to paraffin samples of MIP. And because of the way the assay is designed in this step of amplifying a synthetic probe rather than the original DNA, we find that the MIP data on paraffin in some cases, actually in many cases, is much cleaner with less noise than some of the other arrays that are currently out there. Now, CGH, some of those platforms do work well in paraffin. However, one of the downsides of CGH is that you don't get the allele ratio information that you do from the SNP arrays, which can be very, very important. And also, we're finding, as many people know, that there are regions of uniparental disomy, these copy-neutral LOH regions, that is really key to pick up using the SNP arrays.
And can you use whole genome amplified material?
You can, but the results aren't as good. We didn't do it ourselves, but early on, I think it was attempted, and I think the results were a little bit noisy, right? Because you're amplifying DNA. If the DNA is poor quality to begin with, and you amplify it, it's going to become even more degraded or lower quality as you amplify. One of the nice things, again, we've gotten this to work on as low as 3 nanograms of DNA, so even with small amounts of DNA, we don't find the need to do whole genome amplification. I think, though, I would defer to the AFI representatives, and the technicians. They may have a better, more current answer for that. But ourselves, we have not used whole genome amplified samples.
Okay, thanks, Dr. Schiffman.